Hi, I'm Heather James, conference producer at Technology Networks, and I'm here to introduce our next talk, Navigating Spatial Transcriptomics Technologies, Choosing the Right Tools for Your Research. Please join me in welcoming Dr. Elsa Molina, Director of Single Cell and Spatial Omics Core at the Salk Institute for Biological Studies. With international research experience at laboratories in France, Australia, and the USA, Dr. Molina has honed her expertise in molecular cell biology, next-generation sequencing, single-cell genomics, and spatial omics. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. Without further ado, I will now hand over to Dr. Molina to begin her presentation.
Thank you very much, Heather, and thank you to Technology Network for the invitation to speak today. Hello and welcome, everyone. Thank you for attending the presentation on spatial transcriptomic technologies. So my name is Elsa Molina, and I have been leading the single cell and spatial omics core, formerly known as the Next Generation Sequencing and Genomics core, here at the Salk Institute for more than 2 years. The goal of this presentation is to provide you with as much information as possible to help you determine which spatial transcriptomic technology is best suited for your project. So we are going to first review what technologies we offer and run here at the core at Salk. Then we will review the current technologies on the market. We will review what are the main differences between those technologies, and we will end by homemade technology comparison table that I hope will be very helpful for you and your research. So let's start. So the Salk Institute first was established in 1960 by Dr. Jonas Salk, who is the developer of the first safe and effective polio vaccine. So the single cell and spatial omics core at the Salk Institute is dedicated to advancing genomics research by offering cutting-edge single cell and spatial omics services. So our mission here at the core is to support scientists at Salk, but also external scientists and biotech companies by providing comprehensive consultation, expert guidance, and experimental design, and state-of-the-art technologies tailored for innovative research in genomics. So what we currently offer here at the core, so we offer short and long-read sequencing, bulk and low-input library prep, single-cell, single-nuclei RNA-seq, ATAC-seq, spatial transcriptomics, of course, that I'm going to develop in a minute, digital gene expression. This is all our main services. We also offer qPCR services as well as sharing services. On top of all full services, we are also offering instrument and assay training, as well as cell services that are open 24/7 for certain technologies here at the core. So our main instrument that we have at the core are sequencers. So we have plenty of them. We have the NovaSeq X from Illumina, the NovaSeq 6000, NextSeq 2000, MiSeq, MiniSeq. We have also long-read sequencers such as PacBio Revio and Oxford Nanopore Technologies, the GridION. We have also for digital gene expression, the NanoString Encounter Max instrument, and for single cell, the 10X Chromium X. And here we go for spatial transcriptomics. We are equipped with the 10X Genomics Xenium system, the Visgen MERSCOP instrument, and the Bruker NanoString GeoMx Digital Spatial Profiler. Those advanced platforms allow us to offer precise and scalable solutions for single cell and spatial transcriptomics, enabling researchers to achieve groundbreaking insight into cellular and molecular processes. So now let's talk about each of our spatial transcriptomics instruments, what we have done here at the core, on what we have worked and what we like and what we would say like didn't like too much about all of these instruments. So let's start with the Geomics DSP. So we have run so far only whole transcriptome assays on FFP samples and fresh frozen on human and mouse species mainly. I listed here all the type of tissues we have worked on. And what we can say is this is a reliable instrument. We get very powerful data, and usually all the data that we got are confirmed with other assays. This is a very efficient instrument. What we like is also it provides a free cloud analysis software, and we also can remote, we have access in remote with the instrument. So what we didn't like is we have regularly we have software issues. And the selection of regions called region of interest, RIs, are kind of very long. So usually it takes more time on this instrument to do the assays compared to the other instruments. So now let's go with the Xenium. So far on the Xenium, we have run custom and 5,000 panels assay. We've run FFP sample, fresh frozen, and as well as fixed cells, so human neurons. We have run mouse and human species so far. And what we like with the Xenium is we can analyze very large tissues area. It's user-friendly. It's usually fast. It's true that the 5,000 gene panel can take 5 to 6 days, but the small panels, the small gene panels, so less than 500, are usually done in 48 hours. And what we can do is we can do IF after the run. So you can do an IF in your lab or using, for example, the Akoya technology. We have noticed that we got some software issues, and also the instrument currently the panel that 10x Genomics is offering has some limited species. If you have a certain species, you need to work with 10x Genomics and create basically a custom assay with them. But we hope soon there will be 10x Genomics is going to offer more choices. Then the MERSCOPE instrument. So far we have run only custom panels with the MERSCOPE and fresh frozen tissues on mouse and plants. And what we like with the instrument is it's a reliable instrument. The slide preparation is easy compared to the Xenium and the Geomics. What we like is it's also species agnostic. And we usually get a high number of transcripts per cell when we compare to the other technologies, spatial technologies. The limitation are that we are limited with the number of samples per run. The analyzable tissue areas is kind of small. The custom panel design process is kind of tedious. And also we've noticed the efficiency of the technology is down when we add some protein in the assay, even if you had one protein. And the preparation, the slide preparation is kind of complex. So now let's review the current leading technologies. So we have the 10x Genomics Visium HD, NanoString Geomics DSP, Vision Merscope, NanoString Cosmix SMI, 10x Genomics Xenium, Curio Bioscience, CurioSeeker, and Tracker. Also the Complete Genomics Theomics with StereoSeq. And the brand new one on the market is the G4X from Singular Genomics. So I'm going to give you like a quick review of all these technologies. Of course, this list is not exhaustive, and because the spatial omics technologies would say this field is growing fast, there might have on the market right now maybe some technologies that I didn't put here, but I would say this is the most promising technologies that I see coming next year. So let's talk about like the differences. So the main differences between those technologies are first the single cell spatial transcriptomics versus whole tissue spatial transcriptomics. So we are going to review first the whole tissue spatial transcriptomics technologies with the Geomics DSP from NanoString and the Visium HD. So first question that you should ask yourself is, for your project, do you really need a single cell resolution, or do you need whole tissue? Or maybe you need both. So if you need the whole tissue, let's discuss your choices here. So you will have the choice between the Geomics DSP, which uses high throughput next generation sequencing and a digital barcoding approach to provide gene expression. It leveraged fluorescency labeled probe to identify RNA or protein markers within specific tissue region that we call, that I said previously, RI, so region of interest. Regarding the Visium, is based on spatially barcoded tissue slides that allows for whole slide RNA profiling. The tissue is placed on a slide with an array of spots that each contain a unique barcode. So to summarize everything, I've made a table for you. So where I sorted first, so the type of faces, so it's going to provide you, of course, as we just said, the whole transcriptome, so with more than 18,000 probes. So what type of sample you can use? So for both, you can use FFP and fresh frozen. However, for the Visium HD, 10x recommends more FFP than fresh frozen. So the technology, as I said previously, they are different. And can you use standard slides? So for Geomics, this is actually like a standard microscopy slide that you can use. For Visium, this is a specific slide. So keep in mind that the Visium slide, you need to purchase it, and it's of course more expensive than the Geomics slide that you are going to use, as it is a normal microscopy slide. Then the spatial resolution. So for Visium HD, it's going to be 2 micrometer capture tiles. For the Geomics, it really depends on the region of interest that you are going to draw. Usually it's between 10 to 600 micrometers in diameter. The species that you can run human, mouse, non-human primate, and rat on Visium HD. With the Geomics, you can, it's pretty broad, and you can run, they provide panels for human, mouse, and canine, but you can also work with them if you have other species that you would like to work, and they can make custom assays. Regarding the tissue thickness, you can run between 5 and 10 micrometer with the Geomics DSP. And with the Visium HD, it's between 10 and 20 micrometers. Both are doing, of course, RNA, but you can also do protein with the Geomics DSP. And it's not on top of the RNA. This is a different assay. So you're going to choose at the beginning if you're running RNA or protein. And do you need a dedicated instrument? So yes, for the geomics, because this is the instrument. For Visium HD, you can actually use only protease-only slides, and then it's only a reagent for fresh frozen. But if you have FFPE tissue, you will need to purchase the site assist to transfer your tissue on the slide. So the difference between Visium and Visium HD. So Visium was the first version of the technology and was released in 2022. At that time, so with the first version of Visium, you did not need the site assist. And now with the new version, with Visium HD, released in 2024 this year, you need the site assist if you have FFPE tissues. And finally, the cost. So I know like many of you are interested with the cost, and basically every user keep asking me, what is the cost? What is the cost? So basically the current price, say right now, it's approximately $4,000 per slide if you include the sequencing. So for 10x genomics, it's approximately 3 times more. So this is a very expensive assay compared to the genomics. But again, it depends on your project and your research question that is unique to your research project. So now let's go back to this slide where we are comparing the single-cell spatial transcriptomics technologies with whole tissue spatial transcriptomics technologies. So now let's talk about the single cell. So within the single cell, how to compare them? So there are main differences. So you have the targeted panels versus the whole transcriptome. So the MERSCOP, COSMIC, and Xenium are not doing whole transcriptome, maybe soon, but so far you cannot do it. Curio, StereoSeq, and G4X are providing whole transcriptome. And some technologies can run and detect proteins on top of targeting transcript, such as the MERSCOP and the COSMICs, or you can even do spatial proteomics such as COSMICs, StereoSeq, and G4. So as you can see, this is kind of complex, all the specificities that those technologies can do. So I basically created a comparison table for you to better define what you need. So before going to the table, we are going to talk about a little bit about the G4X. So this is a brand new technology from Singular Genomics that is very promising. So basically, with the same instrument, you can do next generation sequencing and spatial transcriptomics and proteomics, so basically multiomics, with RNA, protein, H&E, everything on the same slide. Also, the number of samples that you can run, as well as the analyzable area, seems pretty big. So this is a promising technology, and hopefully once we see more project done with this technology, I will be able to add this technology in my next table. So if we try to sort the technologies as the previous table as I showed you for whole tissue spatial transcriptomics, so regarding the transcriptome, as I said previously, you have StereoSeq and Curio who are running whole transcriptome. The Xenium, COSMICs, and MERSCOP have targeted gene panel. StereoSeq and Curio are sequencing-based technology, while Xenium, COSMIC, and MERSCOP are imaging-based technology. So what type of sample you can run? Most of them you can run FFP and fresh frozen. However, for Curio, you can run only fresh frozen at the moment. Maybe next year they will be able to provide SS for FFPE. What is interesting is you can also run fixed cells with the Xenium that we actually tested here at the core with human neurons, and it worked very well. For just note that for stereo-seq, for FFPE, it's called stereo-omni, so maybe you have heard about it. So this is the same thing. This is a stereo-seq, it's just with FFPE, it's called a stereo-omni. The resolution, so nearly all of them are subcellular, except Curio, this is single cell resolution. Regarding species, what we like with stereo-seq, Curio, and Merscope is it's agnostic. For Xenium and Cosmic, it's mainly human and mouse, but you can work with 10x Genomics and NanoString if you have other species that you would like to run, but basically would work if you have a good transcriptome reference. Tissue thickness. It's pretty broad based on the technology. So for example, for Cosmics, you don't have the choice. This has to be 4 micrometer thickness. For other technologies, for example, for Xenium and stereo-seq, you have to be within a range of 5 to 10. And what we like is with the Merscope, you can go up to 12 micrometers. So when you're working, for example, with organoid, that might be interesting for your project. Analyte. So of course, all of them are doing RNA, but some can do on top of RNA, you can run some protein. So with the Merscope is you can add protein on top of your RNA, so up to 6. But as I mentioned before, that is not really recommended because the efficiency of your assay is going to drop. For the Cosmics, you can run RNA or protein, so you cannot run at the same time. And for protein, you can run up to 76. For Xenium, what we like is you can do RF post-run with the Xenium slide. So with the same slide, you run the transcriptome. For stereo-seq, what we like is you can do RNA and protein at the same time, so more than 100 of them. And currently, Curio, Seeker, and Tracker, you can run only RNA. So now the imaging time. So this is something to consider, especially if you have many samples. So with spatial transcriptomics, it's more the area, so the size of the samples than the number of samples that you have. So you will understand soon. So here I put the imaging time per centimeter square. And as you can see, there are big differences between technologies. And here I put the analyzable area per run. Basically for Merscope, you can run only 1 slide per run, and 1 slide can contain only 1 centimeter square of tissue. So you can put many tissues as long as the total area is 1 centimeter square. So it can take, depending on if you run some proteins or not, it can take up to 36 hours to analyze 1 centimeter square of tissue. But for example, with the Xenium, you can run 4 centimeter squares of tissue per run, and it will take only 12 hours to detect 1 centimeter square. So this is quite fast compared to the Merscope. The Cosmic is pretty long, as you can see here, because just for 1 centimeter square, it can take up to 78 hours. Also, it depends on the number of genes that you are targeting. So of course, if you are targeting 500, it's not the same as you are targeting 6,000. So this is why it takes longer. And finally, the price. So again, depends how many genes you are running, the number of sample, the area of your tissue. But I've tried to make an average and to estimate the price. So for CurioSeeker and Tracker, it's approximately 3K. However, consider that after performing Curio, you have to proceed with either 10x genomics technologies, single-cell RNA-seq technologies, or BD, single-cell RNA-seq technologies. So the cost of those downstream assays are not considered here. For StereoSeq, they offer service only, but I think they have just started to sell the product. So it's approximately 10K overall. For the Xenium, it will cost you approximately $8 per gene. For the Cosmics, it's less expensive, and the Merscope and the Xenium are approximately the same price per gene. So with this, I would like to emphasize that your project is unique, and especially your research question. So it's very important that based on your research question, you determine the best technologies for your essay. And I hope this table is going to help you. Again, core directors and core manager are the best person to ask and to advise you before running an essay. As you can see, those essays are not cheap. And so this is important that you run the correct essay the first time. And with this, I would like to thank you for attending the presentation, and I'm happy to take some questions.
Lovely. Thank you so much, Dr. Molina, for that fascinating presentation. I'm pleased to have you here with me so that we can take some questions now. We've had a few great questions come in, so I'm going to start with the first one for you. And that one is, why choose subcellular resolution over whole tissue resolution and vice versa?
Yeah, thank you very much, Heather. So this is a great question. So this will depend actually about your research question. So do you need to understand the transcriptome or the proteome at a subcellular level, or do you need to understand that all over your tissue? So it really depends on the research question. So whole tissue, spatial transcriptomics might be only enough for some research question, and sometimes you need a subcellular level.
Excellent, thank you so much. I've got a really interesting question here from Quayle that is, what are the key considerations when selecting spatial transcriptomics tools for oncology research, particularly in understanding tumor microenvironments?
Yes, so great question here too. So what I usually do, because we have here a lot of spatial omics technology here at the Salk. So what I usually do is I always talk and speak with the researcher about the project and what research question he or she needs to answer. So there is not for me, there is not a best tool right now. The best tool depends on your own research and the question that you are trying to answer while running spatial transcriptomics or spatial proteomics. So for oncology, that will depend if, of course, which species. So is it directly human or mouse? So we will start from that, from the species, and then again, the tumor. So the size of the tumor, like the area, how many samples we need to run to answer the questions. So we will start from that. And then after answering all the questions, then we can define the best tool. But I think all the tools right now that are on the market, they can fit, one specific need.
Lovely, thank you so much. Following on from that, then, we've got another question asking, in your opinion, what is the most reliable spatial technology out at the moment?
Yeah, so this is a great question. I would say based on our current experience, there is not really like a reliable instrument. All instruments have their limitations. All instruments have their own issues. We have to keep in mind that spatial transcriptomics is kind of a new technology. It has only like a couple of years. So I really think all the instruments need improvement, especially regarding the software. So I would say that for me, there is not like a very good, reliable instrument right now on the market regarding software.
Excellent. Thank you so much for that comprehensive answer. Another question that we've had in is, how do you see the price of spatial omics technology evolving in the future?
Yeah, so I really hope, like the sequencing price, I really hope the price will decrease in the future, because it's still, it's not cheap to run spatial transcriptomics or spatial omics. So I really hope all the companies will work on that and make an effort to decrease pricing in the future.
Wonderful, thank you so much. And another question that we've had here is, why choose spatial transcriptomics over single-cell RNA sequencing?
Yes, so that will depend on your research question. You might not need spatial. You might not need to know, where which cells is expressing that, or in all over your tissue. So that will really depend on your project. Some people are coming and would like to run spatial transcriptomics just because this is a trend. Okay, so you should never run a technology because it's a trend. You should run a technology because it's going to answer a specific question for your research project, and you will understand better the genetics or the genomics or the transcriptome or the proteome of your tissue or your cells. So this is two different technologies, and you get different answers from single-cell RNA-seq and spatial transcriptomics, or single-cell spatial transcriptomics. So the best way is like to talk directly to the core manager or the core director, or the person who knows about the technologies and who knows the difference, and discuss like fully about your project with this person to determine if you just need single-cell RNA-seq, or if you need spatial single-cell transcriptomics.
Lovely, thank you so much. And a really great question that we've had here, I think will tailor to your experience with so many different technologies, is how does sample preparation impact the quality of your spatial transcriptomics data, and what are some best practices or tips for the different tissue types?
Yes, so great question. So the preparation of your samples will definitely impact the data of your run. So best practices is always to follow the protocol of the specific technologies that you are going to run. Because what the limitation we have encountered so far here, it's sometimes we are running type of tissues that the company hasn't, would say, run a lot yet. So when, for example, when we started running cells on Xenium, we of course contacted 10x Genomics to make sure we were not going to make any, mistakes, or we were going to make sure we wanted to have the best practices for running cells and not tissue with the Xenium. So, but sometimes, when it's new, and you do not really know, even the company doesn't really know what you are going to get at the end. So in that case, yeah, you can try and new things and not following exactly the protocol. But so far, I would say stick exactly to the protocol, because I've seen some people not following exactly the protocol and not having actually data at all, just because they didn't use the correct buffer, for example. So this is very important, and you should always make sure the tissue prep, even cutting your tissue, it's done by someone who has experience and knows how to do it.
Excellent, thank you so much. We've had another question that's come in that's a bit of a follow-up onto the one of why choose spatial transcriptomics over other methods. And that is, can you easily integrate spatial transcriptomics with other omics data types, such as the single-cell RNA sequencing or proteomics?
Of course, yes, of course. So, you should always confirm your data with another type of technology. So if what I would recommend is actually go first with whole tissue spatial transcriptomics, then get a specific list of genes, and then move to single cell spatial transcriptomics. You can also run a single cell RNA-seq to correlate the data that you got with spatial omics. So yes, of course, you can, of course, combine, and I would recommend doing that.
Lovely, thank you so much. We've got time for a few more questions here, so please do send in your questions for Dr. Molina, and we'll get to as many as we can. Another great question that we've had here, obviously you've compared a lot of different technologies. Do you think that there is an opportunity in the future for standardizing spatial transcriptomics protocols and data analysis pipelines, or do you think that will stay sort of quite separated, dependent on the technology that you're using?
Yeah, that is a great question. So currently, the challenge is the analysis for spatial transcriptomics, for single cell spatial transcriptomics. I haven't heard like a common script or common pipeline yet for any of the technologies. So I think there is like quite some work to do. So if you are a bioinformatician, go for it. Please try to find a common pipeline to analyze the data, but that will be great. I mean, it's what everyone is looking for. I mean, at least all the biologists and people who are not bioinformaticians are looking for that. We are looking for to hear about like a common pipeline to analyze those data. Yes.
Lovely, thank you so much. Another great question that we've got here is, what is the current limit of spatial resolution in spatial transcriptomics, and can you see that improving in the near future?
Yeah, so the limitation of the resolution will really depend on the type of technology. So I cannot really, talk about a specific one, but some researcher, depending again on the research question, might not care if the resolution is not too deep. You know what I mean? Sometimes they just need like not a very deep resolution, and some researchers, they really need to know like inside the cells, transcript if they're in the nucleus or not, or in the cytoplasm. So it really depends on the research question, but I really hope that we will have a deeper resolution. What I think matters, based on my experience here at the Salk Institute and all the research projects we have been working on, it's not the resolution, it's more the data, are they reliable and true? So I'm talking about like, the number of transcripts you get and the differential expression between genes in single cell spatial transcriptomics data. So I think I would like more the companies working on reliable probes or reliable single cell spatial transcriptomics data instead of resolution. But maybe, I'm not an engineer specialized in this type of technology. Maybe the resolution is correlated with the reliability of the technology. So I'm not quite sure. But for me, it's more important to know if the data are true and if those genes are really expressed in this type of cells and etc., instead of like how deep I can go within the cell.
Excellent. Thank you so much for that comprehensive answer. We've got time for just a couple more I think here. And I've got one that's a bit of a technical data question, so forgive me for this one. But how do you normalize spatial transcriptomics data to account for any technical variations and batch effects?
Yes, so unfortunately, I'm not the best person to answer this question because I'm not a bioinformatician. So I know that you have different technique and you can normalize with different ways. When you want to compare the same data with different, for example, if you have run the same tissue on different platforms and then you want to normalize the data, I think you should normalize based on the area, because you can, as you've seen in the presentation, you don't capture the same area on different platforms. So you should definitely consider the area and also the thickness, because it's normal to have more transcript per cells and more cells too in 10 micrometers tissue and compared to a 4 micrometer tissue, So you need to think about that when you see people like telling you the number of transcript per cells, the number of cells captured. Yes, but what was the thickness of the tissue, So this is also like what was the number of gene you run. So you need to consider that.
Wonderful. Thank you so much. And I think that we've got time for just one last one here, and I think this is a nice one to end on. So that is, what do you see as the most exciting application for spatial transcriptomics coming up in the future, and how much impact do you think that will have?
Yeah, so what I really would like to see is like more reliable. So like we are sure those transcripts exist, faster, definitely faster, because sometimes it takes, it's very long, to analyze like a big piece of tissue. So definitely that is more exciting for me, like the area and the is it true? Can we trust the data? So this is for me like main questions.
Wonderful. Thank you so much, Dr. Molina, for your insightful answers. I know that we had a lot of questions to get to there, so we really appreciate your time and expertise. But unfortunately, that's all the time that we have for you today. Any questions that we didn't have time to get to will be sent to Dr. Molina after the event. And as with all other talks in the event, this talk will now be available to view whenever you want for the next 30 days. And that just leaves me to say thank you so much again, Dr. Molina, and we'll be back soon with the next talk.
Thank you.